And now, ladies and gentlemen, let's get ready to rumble!
Prophase. Centrioles move apart, chromosomes shorten and thicken, and start to become visible. Nucleolus begins to disappear. Metaphase. Nuclear envelope has disappeared. Replicated chromosomes held together as centromere align on equator of spindle at the metaphase place. Anaphase. Centromeres split and daughter chromosomes begin migration to opposite poles. The two sets of daughter chromosomes approach the poles, cytokinesis begins. Telophase. Nuclear envelopes begin forming, chromosomes begin to become extended and less visible. Nucleolus reforms, cytokinesis continues. Meiosis 1. Prophase 1. Leptonema. The extended chromosomes begin to coil and become visible as long, thin threads. Once a cell enters leptonema, it is committed to the meiotic process. Zygonema. The chromosomes continue to condense, and homologous chromosomes bind each other and begin to align to each other in a process known as rough pairing. Pachynemia. The aligned homologous chromosomes become much more closely associated. This process is known as synapsis. Synapse chromosomes may undergo crossing over in pachynemia. The chromosomes continue to condense. Diploidema. The homologous chromosomes of each tetrad begin to separate, but they remain connected at points of crossing over. Each point of crossing over is known as chiasma. The nuclear envelope begins to break down. Dikinesis. The homologous chromosomes separate further and the chiasmatic terminalizes. The nuclear envelope has completely disintegrated by this stage. The centromeres of the chromosomes become attached to spindle fibers. Metaphase 1. Assembly of spindles is completed. Each chromosome pair aligns across the equatorial plane of the spindle.
She's the reason it happened, but she's overreacting, and it's all because she don't want things to change. So cry if you need.
Anaphase 1. Dyads separate and migrate toward opposite poles. Telophase 1. Chromosomes each with two sister chromatids complete migration to the poles, and new nuclear envelopes may form. Meiosis 2. Prophase 2. The nuclear envelope is again dissolved and the spindles is set up again. Prophase 2 is identical to prophase of mitosis, except that there is half the amount of chromosomes. Metaphase 2. Again, the chromosomes move into the center and line up. Now there are two chromosomes instead of two tetrads, so that the chromatids will split off this time. Anaphase 2. The kinetochores move towards the poles, splitting up the sister chromatids. Telophase 2. The chromatids concentrate in the poles, and the nuclear envelope is reformed and the spindle again is dissolved. The cells divide for the last time, leaving a total of 4 haploid cells, which have half the chromosomes of a diploid cell.